Welcome to The Chain. The Chain explores the lives, careers, research, and discoveries of protein engineers, scientists, and biotech professionals. In today's episode, we bring you a panel discussion from the most recent PEGS Summit, exploring near-term challenges for AI and ML in biotherapeutic R&D.
Welcome to this panel discussion on near-term challenges and opportunities for AI, ML, and biotherapeutic R&D. My name is Peter Tessier from the University of Michigan. I have the honor of chairing this impressive panel here today. That our goal is to highlight for you some of the areas we see AI and ML really succeeding and impacting our work today, and also where we see the challenges and where we're going to push ourselves and you to focus in the future where we're going to put resources and time and effort to make the biggest impact for the field. So before I get going, I'm going to briefly introduce this distinguished panel. First, closest to me here, Max Vasquez from Adimab. He's the Chief Computing Officer. And next is Vincent Ling. He's Chief Business Officer at Morphocell Technologies, and he's also a consultant advisor for the Bill and Melinda Gates Foundation. Next is Sarel Fleishman, who's a professor at the Weizmann Institute and also chief scientist at Scale of Biodesign. And after him is Arvind Rajpal, who's senior vice president at Xaira. And then finally we have Kadina Johnston, who's a senior specialist in Discovery Biologics at Merck. It's an impressive combination here of people in academia and industry, and I think you're really going to enjoy interacting with these distinguished people today. So I want to get us started here, and I'd like the panel to comment on, for all of us in the audience, a lot of us are antibody engineers, protein engineers. Where do we think, where do we see AI, ML methods making a positive impact in our work today? Like, essentially, where are you seeing the successes in this field right now? So maybe, Kadina, could I start with you on the end?
Sure. So some of the things that we have at Merck is we do developability prediction with ML. We can see a lot of success in that. We also have some methods for humanization that were published out of our Prague site that we see success with as well.
Okay. So building on Kadina, does anyone want to comment on that? Who else? Arvind?
So humanization, affinity maturation are the usual places where I see immediate benefit. And then if you want to add to it things like polyspecificity, polyreactivity, I think those are emerging areas. Those are, I think, probably the places where people want to start with, and that's where, I think we'll get the most bang for your buck starting.
Okay. Anybody else have a comment? Not everybody has to.
So I think that the most obvious impact is through AI-based structure predictors, which are enabling us to, essentially for any protein of known sequence, to generate an anatomically quite good model, and that's enabling more traditional, let's call them, computational design methods, atomistic design methods to take on from there without requiring a crystallographic structure. So that's dramatically accelerating protein optimization and also reducing the need for crystallization.
Yeah, I think kind of related to what Kadina talked about in developability and also we at Adimab have been accumulating data for many years on several aspects of developability, or what somebody calls pre-developability. Some of this sort of easier assays like polyreactivity, hydrophobic interaction chromatography, etc. And also during the effective campaigns, we get an idea where in Adimab we have synthetic repertoires of antibodies. And so we have, and these are designed initially with just trying to replicate a human repertoire. But over the years, we have combined the data in terms of what comes out and also what those molecules, how those molecules behave in different properties, apart, of course, from their individualities in terms of specificities or biological activities, and try to feed back that information into new libraries, new repertoires that we use. And in the first few years, that was all very bioinformatic, kind of mechanical, simplistic. But in more recent years, we've been able to start leveraging some more of the machine learning, AI technologies to design the next generation repertoires.
Okay.
Yeah, so my take is much more of a generalist view. I spent many, many years in large pharma doing search and evaluation. And at the end of the day, the 800-pound gorilla in the room really is, does this make $1 billion drug, right? Even $100 million drugs are not worth pursuing. You need $1 billion blockbuster. And so anyway, from a pharma perspective, you can move the needle to more probability of success is a good thing. So I think it's good to think about how AI impacts our daily work, but we have to remember it works in conjunction with the rest of the company too, right? So it's not just AI for structural biology. The area that I was into is formulation and drug delivery. There are applications there that can move the needle. For example, if you find a method of using AI to cross the blood-brain barrier, that's $1 billion blockbuster right there. Right? It doesn't necessarily have to do with structure. It could be other things that enable a drug.
Okay, well, thank you. So we're gonna approach the elephant in the room here, and I think everybody knows that elephant in the room is de novo design. So what I'm going to ask the panel to do, which is not going to be easy, so I'm going to ask you to do this, is I want to set aside comments about hype and sort of that, which is something we'll get to. What I want to hear from the panel is, for this area of de novo design, what is the best argument for it, right? If we're going to spend some time in this panel talking about it, what are the best arguments for de novo design? Anybody want to take that one?
Yeah, I can start. So I spent about over 2 decades using traditional methods to make antibodies against disparate antigens. And in general, things work. It's a very successful field, and we made several drugs, and antibody engineering and antibody drug discovery is a very well-established approach to making drugs. But throughout the years, there were always some targets that we wanted to go after that we were not able to go after or make satisfactory molecules. Because traditional methods just give you whatever is immunodominant or whatever shows up on the display methods. You often just get what the system gives you and you make the best of it. And we've been fairly successful doing that. But the what if for me is, are those targets that we worked on for many years that we thought were biologically validated, were fundamentally great targets to go after, that we weren't able to drug? So that's a great use space to go after, and then to use machine learning to do de novo design. What I'm not a huge advocate for is using machine learning to speed things up, because in a 10-year trajectory of discovery to development, shaving 3 to 6 months is probably not going to be as impactful. I think the key for me is to focus machine learning and de novo design to identify intractable epitopes or epitopes that would be very difficult to go after using traditional methods, or where you got molecules that were suboptimal.
Okay, and maybe can I'm going to ask for other feedback here from the panel, but can we get a little more practical? So when we think about what are these maybe hard targets that would justify de novo design, could the panel comment on what are some of those? Maybe Sarel?
So I think that Arvind's framing is absolutely correct. The main question for de novo design is, how do you target difficult epitopes? But there is also an inherent challenge in this, in the fact that many of these intractable epitopes are highly polar and also concave. And often what we can do with de novo design is because de novo design really, the approaches really put a high premium on foldability. That means that you mostly get alpha helix bundles, which tend to be quite convex. And that's very difficult to find binders for these deep pockets and polar pockets in proteins. So I think this is very true, but also very difficult. And I think that if de novo design really to become very impactful, we need to move beyond alpha helix bundles to more complex topologies, protein topologies, such as the ones we see in antibodies, actually.
Interesting. How about maybe just keep pushing a little bit? So what are these targets that the field should be going after? What are these high value targets that you see, even classes of proteins?
Yes.
What are the greatest unmet needs right now that might justify this field?
So GPCRs, ion channels, those are incredibly difficult targets.
Okay.
So I think, right, that would be, yeah.
Or epitopes that are, shared epitopes between two molecules that you want to hit at the same time, where it would be difficult to immunize or difficult to raise a molecule against using traditional methods. Things like that.
Yeah, I think places where you have something about the biology, or along the lines of what Arvind just said, about you want to hit target A, B, and C, but not D and E from the same related family. So that usually, although the way the problem is formulated, it's not always necessarily this is the epitope, but by the time you look at the sequences and the structures, it really boils down to very, very narrow areas. And although traditional methods often can do that, especially use display, but the truth is sometimes it can be time consuming or maybe not succeed. So having a way to molecularly target that, it would be fantastic. It could move the needle. Maybe not $1 billion, maybe two. Who knows?
Yeah, corollary to what you're saying, Max, is going after targets that would be conserved across species, because you need species cross-reactivity for safety and efficacy and things of that nature. And quite often, that becomes a challenge, and then you have to go after surrogates, and that just becomes like a 2x discovery program.
It seemed like in our discussion, we did do some homework for this, so the panel has been working on thinking about this. It seems like the themes we kept hearing was, it's not about speed, which I think you commented on, and it is about the value of de novo design would be when you have high confidence or relatively high confidence in some type of mechanism or epitope or application or conserved region across multiple things. It seems like those things then motivate you, or the targets are difficult. Am I getting that right, panel?
Yeah, okay.
Please.
Yeah. So let's cut to the chase. At the core of it, it's the target, right? And so creating antibodies, I've done this before too. I'm a biologics person in previous life. So the idea is that you start a biologics campaign once you know the phenomenology that you're going after. So typically you have some disease model, they find some enzyme or some protein, some surface protein that might be responsible for it. Then you have an antibody campaign. So within that context, AI is a tool, right? There are many ways of addressing, problems, but AI is part of that. I think if we invert the equation, you say, okay, well, how can AI modifying structure create a new therapeutic? I'm not sure the ball rolls that way. It goes the other way. And so it's always going to be in the context of the larger scheme of biology.
Okay, well said. So what I'd like to do is transition to the problem of this field, right? And so I think that when we discuss as a panel, it's not surprising there's hype in this field, given how valuable this potentially could be, right? Chat GPT generated molecule that does exactly what you want and all of these things. I think we understand that. We understand the commercial significance here. We understand the potential. I think we've just motivated some of the reasons why you'd want to do de novo design. But because of all this and this intense pressure, this has also led to problems of hype and sort of almost violating the scientific method. And so the question is, what do we do about that? If we all as a community care about AI and ML advancing the field, we all want to see this flourish. Really, the question is, what do we do? What is the path? And I'd like the panel, can you provide some recommendations or thoughts about how to advance this field given that it's very, very high potential, but there seems to be a gap between what we want and what we imagine it can be and where we are today. And what does the panel advise this community? How do we move forward here?
Can I start that?
Please.
Okay,
Sure.
so from my experience in a pharma company, there's two levels of looking at AI. Let's say the work in a company is like a bell-shaped curve where there are people who are productive and there are people or groups that have the potential of finding a billion-dollar drug. And it's always framed in terms of a billion-dollar drug, $3 billion drug, in fact. That's what you need these days. So if you use AI so that the core population of your company is moved 10% more efficiently, that's a significant gain for the company. So when people say, let's use AI, it's not just for protein design, it's for use for the entire company. How can you apply AI to shift the median curve, the median over to the right so that you have a more productive company and hopefully you can accelerate faster. Now, the people at the very edge in discovery, if you're going to say AI, it better address how do you increase the probability of success so it's beyond 5% for oncology or 10%. If you can't get beyond that, then AI is no use, right, for that type of people, right? But for the general population, if you put the pieces together, it can make a difference, right? So it depends on how you view a digital strategy in a holistic view.
Right. Yep, I think that's good. I think, you're keeping us honest at a higher level. I think, maybe taking it down though for a minute to that lower level of protein design. Maybe Sarel, could you get us started?
Right, so for protein design, my sense is that benchmarking is critical. And currently many of the papers and claims being made in the field are not experimentally validated to the highest standard, I would say. I think Andrew Badbury's initiative to start benchmarking these methods really explicitly in his hands, so one pair of hands. It's all outsourced, fine. But in a very principled way, at least. And I think that that's really the way to go. I'm sure this will be a work in progress because we'll understand what AI and other methods are capable of doing and what they're not capable of doing. And then this will probably evolve. I hope it will evolve over time and that this will be maintained. So, but that I think is really essential because currently, I think that many of the papers are not substantiated in this way.
Okay. Yeah. Kadina, could you comment? And could also the question, what do we really mean when we talk about benchmarking? What are different ways you'd imagine that?
Yeah, so I think what I was going to maybe elaborate on Sarel's comments was a lot of times when we set up an experiment, we have a limited amount of molecules. And so we want to test our new methods. But I think we have to remember, like, maybe half of those molecules can go to our new method, and the other half should go to that standard way that we would typically engineer that molecule. And I think that can be, if you're an ML scientist or a computational person, really connecting with the wet lab scientists and really integrating your groups such that your protein engineering teams are not just wet lab scientists, they're not just computational scientists. So making sure that you're really providing fair comparisons between those methods. And then I think you can start to find ways where maybe you use some wet lab scientist intuition combined with some machine learning, and you can get even better molecules with those sorts of attempts.
Yep, I agree with everything that's just being said. I think this concordance between, building the concordance between in silico metrics and actual ground truth is vital. I think quite often we see computational teams come up with de novo designs of molecules and publish or share in silico metrics and think that that is, the end of the experiment. When in fact, I think that's the start of the experiment. When we need to make those molecules and really test them and see what is the concordance between those. And sometimes there is concordance and sometimes there isn't. And especially when there isn't, it's a great learning moment to understand what parts of the model need to be further modified or refined. So I think that's absolutely needed. I just wanted to take a step back to what Vincent said. I think AI and machine learning can have a very broad impact, not just in molecule design, but also in biology and also in clinical development. So you're absolutely right that we have to keep that in mind. But given the audience, and I think most of you guys are probably protein engineers and are protein and or antibody engineer and discovery centric. I think in discovery, when you look at de novo design, it's fairly successful in helical bundles and making molecules that are, where the physics has been pretty well established and the models are pretty sophisticated in making binders. But those helical bundles are not easily made into drugs. So my recommendation to the field and everybody would be to start moving away from the helical bundles, use the helical bundles as reagents, but try to emphasize and try to understand what does it take for a molecule that comprises of mainly beta sheets and beta hairpin loops, like antibodies, to understand the physics behind those, to understand the machine learning algorithms behind those. I think that's where the field needs to go. And it's going to be a lift. But until we do that and initiate a proper architecture around that, using something like what was used for CASP and protein folding to be able to benchmark molecules that are then designed and then tested in the lab, I think it'll be difficult to move the needle on it.
Just a comment, and this is not my personal view necessarily. I tend to agree with you. However, if you spent time yesterday at this conference, probably in this room, there were lots of people doing mini proteins, many of them helical bundles. And as far as I can tell, they want to make them into drugs. So I think, just to throw it out there. People, I mean, yes, it's true. It seems like the design problem is more tractable for those. The flip side is that those going to be real drugs. Well, I think that's something that remains to be seen. Although I tend to agree with you.
Sure. Yeah, I think there's levels of risk, right? And some of these non-standard formats. You know, I think the issue around benchmarking, maybe just to put it in a broader sense, I mean, really, what does that mean? So I think the panel discussed, okay, benchmarking, first of all, fundamentally, the panel said it's got to have a very strong experimental component. And I think that there's concern from the panel that the field is moving so fast that actually they're almost moving away from experimental validation, which is not good, right? So I think benchmarking, we need stronger experimental validation. That has to be, you know, we have to remain true to that, right? I think that was one, Vincent.
Yeah, so maybe if I can draw an example outside biologics, go back to, you know, drug delivery technologies, which I'm very familiar with, right? And so, for example, I can think of 3 companies immediately that use AI. So Mana Bio in Israel, they make LNPs, and they use computational AI methods to make better LNPs for targeting and all that. And it's iterative. So they make a design, they test it, and they go back through the loop, right? There's Nanite that's in town here, and they do the same type of thing except with polymers, not lipids, right? They go through an iterative thing, and they feed on itself, and you create a loop. So you generate data, and you go back and you create new molecules, and it's self-contained. The third one just got $7 million in Series A just last week. It's called Intrepid Labs in Toronto, and they do small molecule formulation, and they have an array of excipients that they mix together in different ratios, and it's all automated. And so you can give them the target or your, in this case, small molecule that you want to formulate. It gets results from their initial set, and it deviates from there and creates another set. And all this is automated. In fact, all 3 these companies have this automation loop, and they feed back. So in a case like this, the circle is complete. All of them still suffer that they still have to go through an animal to see what happens. But in formulation, your requirements are less, right? You're not looking for target engagement and then having a cure. You're looking for release, solubility, and all these very distinct things. These type of things are very engineerable, very open to being, you know, applied to by AI, ML. And I think, you know, that's the type of thing that we have to think of, you know, these iterative little things that are lower hanging fruits that can make an impact to the field.
Yeah. I mean, I think the panel, you know, was sort of emphasizing, you know, AI and ML for the field, and for us to be experts, of course, any tool needs to be matched with the right problem, right? So I think that the broader context that we're trying to figure out is where is AI and ML best suited and best paired with the problem. So we're going to transition off this de novo design point. I think the only point we wanted to make as a panel is that benchmarking, we talked about experimental benchmarking. And then I think the other part of benchmarking that gets a little fuzzier is benchmarking against the best methods that are close to you, right? Because this field is not being pursued in a vacuum. It's being pursued relative to the best technologies otherwise for generating, for example, antibodies. And so I think there's a lot of different ways the field needs to advance in benchmarking to really prove the value and the significance. All right. The panel felt that we wanted to get the elephant in the room out of the way, so we tried to do that. But where the panel sees maybe the greatest significance overall was not de novo design itself, although it's important, and we tried to address why, but actually optimizing existing molecules to make them better. This, you can think of multi-parameter optimization. We can think of ultimately zero-shot predictions, things like this. The panel has mixed feelings about some of these, but overall, there was a strong sense that this is an area for opportunity now. Now, with that being said, this field has been advancing. It's been going for years now. The question is, what is it practically doing? Where are we succeeding in this area? And where do we see the challenges, and where do we want the field to go in the future? Who wants to take? Sarel, will you get that started?
So I think that it's quite clear already for almost 2 decades, really, that machine learning excels when there's good data on the specific property that we're interested in. So if you have a protein and you have information about the mutational effects on activity, stability, or whatever you're interested in for that specific protein, then machine learning can help you navigate the complexities of combining mutations in order to produce better molecules. There's a related problem of epistasis where the mutations, some of the best mutants may actually involve combinations that are not beneficial on their own. Actually, a single mutation is deleterious, another mutation is deleterious, but combination is useful. That's something that machine learning is traditionally not very good in. Because there's no data on that. This is something Francis Arnold, who's been developing machine learning for a long time, has been arguing that this is an area that's difficult for machine learning methods. The key problem then, and as you see in this example, is that when you don't have specific data on the mutants and their impact on activity, then, which is maybe zero shot or whatever, then it becomes very difficult to use machine learning. And often this is the scenario: we get a new molecule, a new protein, an antibody or an enzyme, and we'd like to improve it, but all we have to go by is its sequence, maybe a structure or a model. How do we improve that? And I think machine learning methods are not suitable for that.
Could the panel comment on if anyone has any practical experience, what kinds of properties, or how is this being useful practically? Could anyone comment on some examples of where they see this multi-parameter or fixing problems in existing molecules, making problems better? Could anyone comment on that?
Can I do that real quick?
Okay.
All right, real quick. So the idea is that once I was approached by an AI antibody company, and remember, if you're talking about the world of commercialization, it's money, right? And so their proposal is that they can take any antibody out there that's been that's a therapeutic, and they can help you break patent, so you can make your biosimilar faster. And that's totally applicable. It could generate billions, right? So to go for discovery might be too hard, but to use it for other means to break patents is an application, and people are pursuing that.
Interesting. Okay. Say that again. All right.
No.
You don't need AI to break patents, apparently.
It helps.
Just talk to Andrew. Okay.
It helps.
But back to sort of practical things. Could anyone comment on where they're seeing this sort of using machine learning AI to improve existing molecules? Any practical examples or areas you would suggest the field focus on?
Yeah, I think the way at least we've been doing at Adimab, it's a little bit different because it kind of matches our technology, how we think about it. But it goes back to the repertoires. We have a very well-defined kind of initial library. So all our hits, we kind of know what they look like. So we can, in some sense, pre-optimize, create like optimization repertoires to improve in a general way. So once we know this antibody belongs to family A, then we have a cassette of optimization libraries for that. And that's something, you can imagine when we're doing this at the very beginning, 10 years ago, that it was a pretty dumb library, N and K at every position. Not exactly AI. But as we learn, obviously there are some things that are clear. And as the oligosynthesis, that's where the convergence of technology starts to help with the advent of the twists and Agilent technologies. Then it becomes a lot easier to go and do that and to use the previous data to refine that. And then so we find that we can optimize faster without getting into holes. It's not exactly multi-parameter, although eventually we have to test it. But that's at least a way. It's not the traditional way, but it's a way in which we have used the technology to help.
I think that's a really important area, right? I think making better libraries that are already being used in normal workflows to make them be more drug-like, and it seems like a very attractive area for this approach. Others?
There are, sorry.
Go ahead.
There are existing, academic groups like Kim Labs or Mani Lab have used large language models to be able to do affinity optimization of molecules or humanization and affinity optimization of molecules. And there are some companies that are claiming that they can do that, like Big Hat and Cradle. So I think there has been progress made in multi-parameter optimization, both in academic and industry settings, that I think people should really take a look at and see if that's a great entree into using AI and machine learning for drug discovery.
And I think building on those kind of lab in the loop methods from Cradle and Big Hat, kind of the opposite side is not just optimizing your single molecule at the end, but can you improve the quality of your initial pool of hits? So starting out with a large pool, at some point you have to make decisions. And so if you can have models that look at your diverse pool and say, these are the molecules that are going to have the best chances of success downstream, and maybe those molecules also go to a lab in the loop process as well, or they go through their standard lead optimization, but then you can increase the chance that any of your 100 initial molecules could be a drug.
Excellent. Another area that seems very logical for ALMI is developability. We've commented a little bit on it, but this field is not new anymore. It's been around for a decade plus. The question is, where are we with predicting developability properties? Is that now a solved problem? Is it making real impact? Do we have any what do we recommend for the future in that area?
There is go ahead.
I can talk
Go ahead.
about it. So I think it really depends on the assay. And I think one thing that we see a lot is you have to think about what noise is in the assay already. And if you're talking to the scientists and they're like, well, this is a really hard assay to interpret, I can't always identify the right peak, I'm not exactly sure if this is the right area, you can already sort of tell that that's going to be a harder problem to work on. Because if you can't be sure that that data is accurate, it's going to be hard to predict as well. So I think making sure you're aligning on the right assays. And I think also asking the question of, do you want to be able to predict on a diverse set, or do you want to be able to predict a mutation that improves that property? They're two very different questions, and the data you collect to do that modeling is going to be different as well. If you want to predict single mutations, you're probably going to need data that shows the effects of some single mutations. If you're going to want to predict on that diverse panel which molecule is the most developable, you're going to need a diverse data set of those properties.
Yeah, if you look at biophysical characteristics, things like solubility, aggregation, viscosity, I think people are making a lot of progress, including your work, Pete. Where I think I worry about and where I think we need more work is immunogenicity, biological developability issues, where we understand so little about immunogenicity. And I think separating those two is probably a good idea, because I think one may be more tractable in the near term than the other.
Maybe in this context, so I think we're beginning to lay out how complex this problem of optimizing biologics to serve as potential therapeutics is. And And I think that there's currently a sense that AI needs to provide a unified monolithic solution to all of these problems, but it excels in pattern recognition, essentially, right? Again, when we have data on the problem, then we can use machine learning and AI. In other areas, it does not really do so well. But there are other computational disciplines that can be combined. And this is something that I've been working on intensely. How do you combine physics-based, let's say Rosetta, atomistic calculations with machine learning in order to provide better predictions? I'll talk about this evening, but I don't want to take too much time. But I think that's really, at least for the foreseeable future, a good direction to pursue.
I can make it. So, one interesting thing about that too is I was looking into some things a few weeks ago, and we're talking biologics here. If you take a look at the small molecule space, right? So that Schrodinger, the docking software there, I mean, that's the tool that the industry uses to create new small molecules because it can fit binding pockets. But if you take a look at the corporate presentation deck they have, they don't even mention AI ML. It's buried in like slide 6 or 7 saying, oh, this is part of our process. But their core expertise is the physics.
Yeah, yeah.
It's the physics that make all the difference. The AI is just a tool underneath that.
Agreed.
That's why it's called Schrodinger.
Pardon?
That's why it's called Schrodinger.
Okay,
As opposed to Hinton.
We're touching on the topic of developability, and part of the common thing that comes up there is data. And so there's a question around, and maybe there feels like there's been some growing interest in recognizing that there is need for large data sets in that field, because just as was shared, AI ML benefits when it can see patterns and when the data sets are sufficiently large. There's been growing interest, it seems, in developing, even sharing data sets, often because this is more pre-competitive like work. It's not necessarily specific to a given target or a specific disease application. So this question we were discussing as a panel keeps coming up. Is this the time, or is there a growing sense, a growing desire? Do we think it's the right time to start thinking about somehow sharing data in the especially in things like developability, although it could go beyond that? For those of us at companies here, you're the ones that have a lot of this data. Thoughts on this? Kadina, maybe starting with you.
Sure. So at Merck, most of the data that we collect is for programs, and I think that can be hard to convince lawyers to release, but we are doing some efforts. So for example, we have a postdoc right now who's working on collecting a VHH data set of diverse VHHs and doing full panels of developability properties on those. I can't promise you that data is going to be public, but because it's not on our proprietary targets, it's more likely that data sets like that could get combined into a larger, especially if other companies are willing to post up their own data sets as well.
Okay.
Yeah, if you go back and think about how we made progress on folding, I think we could all agree that the PDB really helped with trying to solve the folding problem using AlphaFold or Rosetta Fold. Now, we need a similar effort for protein-protein interactions. Now, a good place to start is obviously the observed antibody sequence database. And thanks to the Dean Lab, they've compiled it and made it more tractable to use. So that's a fantastic place to start. But in addition to that, we need vast amount of binder data sets against as many diverse epitopes containing different secondary and tertiary structural elements in order to be able to train models that now understand and can be able to do what's called transfer learning, to be able to make molecules against any epitope. So that's going to be a fairly large lift. And there are folks who are doing it in industry separately, and how to do it pre-competitively together for the whole, for everybody, I think it's going to be a challenge, but that's a challenge worth contemplating and trying to understand how one could do that.
Sure.
In addition to generating large amounts of data, one thing to consider is that people have been doing deep mutational scanning libraries for a very long time. Let's say the insights, the general insights that came out of it are not so great. What we need to do is to figure out a way of generating diverse sequence and structure data for any given problem that we're interested in a reliable way. And that means the challenge here is that if we just do random mutagenesis, we'll mostly get non-functional, dysfunctional proteins. And that's not going to help us learn the rules for designing better proteins. What we need is a way to generate useful data, of course, combining negative data, but also positive data. And that's a real challenge. And again, I think that the combination of methods, sophisticated methods using atomistic strategies to generate sets of data, and then machine learning to train on these data can help us go beyond the current state.
That's great. Yeah, I think that was a thing that we were grappling with, is the fact that it's easy to say you want more data. It's hard to say what exactly the data you want is, and how even to think about designing experiments to generate the right kind of data, which then would be, I think, Sarel, is that what you're hitting on?
Exactly.
That would be most useful for model training.
Exactly.
Right. So that's obviously a hard problem. We're not going to solve that today, but I think that's really a challenge to the field, right? Is to really think critically about what is the data that you want, right? And what is the data that's going to be most useful for meeting sort of the AI/ML application of your interest, whether it's binder discovery, optimization, and so on.
I mean, just a clarification. I mean, I don't know when you talk about DMS. I mean, I do think, although we don't necessarily do a lot of it, I think it's very useful for the problem at hand.
Yeah.
I think it's maybe less useful if you want to generalize it to some random antigen.
Right.
I think there be other data sets, maybe along the lines of what Arvind was talking about, where those data sets could be important. But I think diversity of epitope and paratope, both are important.
I mean, I think in the end, this deep mutational scanning, we get to learn locally.
Yes.
But the problem is it's hard to generalize, right? And I think that's where the challenge. I think if we think of complexity, we've sort of, in my opinion, this conversation so far has focused really on monospecific, sort of conventional antibody binders. But the reality, of course, is a lot of us are being pushed to work on multi-specific molecules where it's more than just one target, or it's more than one epitope, and so on. And, it's a hard problem to think about because we don't agree on formats. There's incredible diversity of multi-specific formats that are being pursued. And, if you think of problems, let's take something where it's somewhat tractable, something like developability. Is there any progress being made in this space of using AI/ML to predict properties, or maybe it's activity or function, or where do we see the field needs to go in this area?
I mean, this is blue sky, but basically going back a little bit, and it's actually talking about the biology. There was this company called Merrimack, and part of their idea was to have computational biology. This was pre-AI. In trying to understand cellular circuits in terms of trying to design, or rather than design, specify what should we hit HER2 and EGFR, or which affinities, and essentially have a model that would do that. You can imagine, and still this is blue sky, with some of the projects going on in terms of the cell atlas, collecting lots of cell data, where you can start imagining taking those models to a very, very different level, where now you're really doing deep learning of that, and then bringing those back and saying, okay, which 2 targets or 3 targets should I hit this particular cells, and even with what affinities or strengths. And then, of course, now you go back, the idealized world, and talk to Arvind and whoever, and who will actually make you the individual binders, or just immunize mice, whatever you want. But in terms of having that big picture of biology. But that's maybe this is not exactly short term. Yeah.
So what collection, sorry, Vincent, so what collection of biological targets to hit to get the effect that you want, but then taking it to the next step where what collection of epitopes geometrically to be able to co-engage. I'm assuming when you talk about multispecifics, you're focusing towards co-engagement as opposed to being able to hit two targets, which you could potentially do with a combination of two molecules. And you're not tied to the fixed stoichiometry of a multispecific. So if you are really looking for co-engagement, identifying the optimal epitopes that give you the right output, particularly if you're thinking about agonists, it's not trivial. It's not a trivial problem to solve. And traditionally, it's been done using experimental methods, empirical approaches, which has been okay. But if you have, let's say, 10,000 molecules against target A and 10,000 molecules against target B, how to find the right combination of molecules to find that Goldilocks combination is not trivial.
Yeah, I can give a concrete example of that. This dates back about 21 years, and I was at Genetics Institute, and I led a program there to look for the next generation anti-CTLA-4. At that time, our company was going head-to-head against Gemalisant in making ipilimumab. So we had our lead candidate, we had backups. It was my job to make the next generation because we knew that just a simple antibody might not be sufficient. And so basically I took our two lead candidates and we fused them together as bispecific single chain. Back then, the word biparatopic hadn't been invented yet. This is 25 years ago, right? And believe it or not, when we put it on the assay, it gave completely different biology than we expected from the antibodies alone. And this is a complete phenomena, right? We didn't expect this. In fact, when I gave it to the cell biology team after the construct, they said, Vincent, your prep is contaminated with endotoxin. We can't, it's giving us biology, a readout that we can't explain separately. We can't, put these separate parts together to get the same result. Something weird's going on. And then I realized, hey, this biparatopic is triggering a different biological pathway. And this was published. But, unfortunately, the reagents ran out. We got bought out by Wyeth, and everything got buried. And so, we're still working on that. I co-founded a spin-off company to pursue this for anti-CTLA-4. So I think there's some benefit in phenomenology and having a keen eye. If our cell biologist said, Vincent, don't work this out, this is just garbage, then this would have never come to light. But it takes that special eye for someone to say, there's something weird going on here we have to pursue. And that's where value comes in, right? It's the phenomena. Then you end up with all the target biology because now you understand there's something to go after.
Just very interesting. Just to bring this back, when we're thinking about multi-specifics and the role of AI and ML, I think we talked about, for example, at the binder level, we were talking about, Arvind was talking about co-engagement of multiple targets. One interesting thing when we were asking this question of what data do we want, right? We're saying it's not just more data, but what data do you want? Interestingly, the panel was discussing they'd really like to have, at the earliest stages possible, epitope information, which when you start thinking about trying to, eventually use AI and ML to predict what's going to work best for multi-specifics, clearly epitope information would be very valuable. Of course, getting epitope information very early, though, is challenging. Panel, am I characterizing that right? Arvind, you want epitope information, is that correct?
Yeah, and with the models as they are right now for structure prediction, if you know the structure of the antigen, having low resolution epitope information may be enough to be able to dock the molecule, to get a sense of what the epitope is.
Interesting.
And that would be experimental information.
Right, and that would be pertinent, yes.
Okay. I'd like to push just a little bit though on this multi-specific thing a little bit more, because the reality is a lot of us in this room are dealing with multi-specific antibodies, and it's a lot of work because of, we said, the combinatorial nature of it. And it's not easy to predict what happens from a expression, developability, ultimately to function. So I still would like to hear more from the panel. Do we see opportunities to improve this workflow? Because it seems very rudimentary right now. It does not seem like seems like there's a lot of opportunity. Could anyone comment? Kadina, maybe you could start what Merck is doing with, multi-specifics in this area.
Yeah, I think the combinatorial problem is definitely still an open question, because by that point, we're not testing a lot of molecules. We test them more at their pieces level. So if it's a VHH, a CFE, or some sort of bispecific, maybe you have data on both of the mAbs alone. And so you can start to predict for each of those mAbs independently, and then you sort of hope that that prediction together can describe what that will look like as a full molecule. One of the things that we're also hoping with collecting our VHH data set is if we learn something about the stability of single like VHH domains, can we translate that at all to VHs of mAbs, VHs of bispecifics, and work more at that single domain level? Can we start to build up an idea of what that full molecule will look like?
Mm-hmm. All right, how about we shift here for a minute and think about, when we think of, AI ML, we talk about data, but then part of data is data generation. And there was comments on things like lab in the loop, which effectively means, where are we as a field in practically, not theoretically, but practically implementing this sort of designing, learning, and, redesigning sort of over and over again. Is this practically impacting the field? Is this, what do we, if it's not, are the cycles too slow to be practically impactful? What do we think about this area? Where are the needs, and where would we recommend people put effort in?
Yeah, this is not necessarily something we are doing, but from the publications and what people talk about, Genentech has talked about it quite a bit in some of the lab in the loop, as well as Big Hat, and there are others on that. I mean, I think the one comment that I can make is that if you spend a little bit of time looking at what they're doing, you realize that it's a very intimate collaboration between the computational part and the lab part. And it has to be very, very compatible. A lot of thought goes into the workflow. Otherwise, it's not going to work. And so, this obviously resonates with what we talked about earlier in another context of the validation and the reality check. And so I think that to make that lab in the loop to work, you really need to have those connections, or at least that's the impression I get. Yeah.
Yeah, so I think in the past, it used to be beyond the range of an academic lab to do these learning loops, but improvements in computational design, of course, but also converging with DNA synthesis. The products we get out of companies such as Twist are much more reliable than they used to be. It used to be very difficult. If we would order oligo pools, they would be not really great in terms of their quality. It would be very difficult to combine them in order to get really the desired and designed mutations from them. Nowadays, this is there's been a real shift and improvement in the quality of these oligo pool libraries and other products from such companies. And I think that now it's still a slow process, but it is feasible now, even for a lab, an academic lab with limited resources, to carry out these learning loops. And it's hugely impactful. I think that we are learning a lot from this.
Yeah, I think something that Max brought up, the design, build, test loop is a multilingual sort of an effort where you have three groups or three areas of work that are trying to work together and trying to make sure that everybody understands each other is actually not trivial. Yes. And then getting people to be able to understand each other so that you're all pointing in the same direction can take a little bit of work.
Yeah, they all have to be in the loop.
Have to be. And after that, I think from an experimentalist perspective, from an empirical side, of course, facile DNA access is a very big part of it. Of course, expensive DNA can add up really fast. So something to keep in mind. But to be able to then make those molecules, either in a segregated fashion or in a library, in a facile manner. So if you want to do a library, doing yeast display, there's a certain timeline it takes to do yeast display. And then to do the NGS analysis and the sequence analysis, there's a set amount of time that
you can do. And you can try to reduce that time, but there's not much room to shave time off there. But the key is to be able to iterate through this fast and multiple times to be able to generate the volume of data and the volume of labels of binders and non-binders to be able to train models. And that continues to be a challenge and continues to be, I think, something that at least we at Xaira are really focused on trying to continue to optimize.
So we're going to transition here. Kent has told me we have about 5 minutes left, and we'd like to hear from you. So we're going to now take questions from the audience. So please, if anyone has a question, could you come up to the mics here in the middle? And maybe while we get people moving, I'm sure they're there. Andrew, you're going to be making your way over, I hope. While we have people getting ready, Andrew will go. Okay. He wasn't on the panel, but he was on the panel. He's here in the front row, so we need to have a Andrew, welcome.
Thanks. So I was on a panel last year, and I don't know whether it's the composition of the panel or the passage of time, but I get a sense at this meeting that the enthusiasm for AI and machine learning is somewhat less than it was last year. I think people are being a bit more realistic. And I would ask the panel, I mean, it seems like there's two things. One is you can use AI and machine learning to improve antibodies. We can do that experimentally. So going to Arvind's point, you're just reducing the time it takes to get to a therapeutic molecule. And within the context of a 10-year process, what does a few weeks or months, what difference does it make? It doesn't make much. The other thing is to look at antibodies that can be doing something new. And I would hope that that's where AI machine learning goes. But if we can continue to generate those molecules experimentally, then is there a role for AI machine learning?
Yep, good questions, very general. Anybody want to make a comment?
Yeah, I can start. I like to look at the field of machine learning and antibody discovery in two separate fashions. One is the de novo design or making of molecules that you couldn't make using traditional methods. And of course, if you can use traditional methods in a facile manner to make molecules, then why not make them that way? But then there's the whole other concept of doing multi-parameter optimization, which traditionally, and correct me if I'm wrong, is actually fairly sequential, where you try to optimize affinity, then you try to optimize developability, then you try to optimize immunogenicity, and hope that you get it all right and make sure that you have a molecule at the end. My hope is that we do multi-parameter optimization in a parallel fashion, that we can optimize all these concepts and these attributes that we want in the molecule quickly in a manner and do them in together so that you get to the end, faster. Because in the discovery pipeline, I think we spend a bulk of our time doing this optimization. And maybe that's where we should put our effort initially, at least as a field.
Okay. Dane?
Dane Wittrup, MIT. I want to thank the panel for just refreshingly measured and thoughtful comments. And Pete, you said, how can we combat, frothiness and hype? If there's any way to memorialize this, a transcript of some sort, MABs, hello, maybe something posted there. And other than just the intrinsic, character of you individuals, I'm thinking, why is it that you're saying things that make me nod my head so much that I'm spraining my neck? I think you're all trying to make molecules. You're not just promulgating your own personal academic or business reputation or trying to make money by knocking off other people's patents or anything. You're trying to make molecules, and you will use the tools that work, and you're interested in finding out what already works. And just love the discussion. Well done.
Thanks.
Right, trying to make expensive molecules, Dane. $3 billion molecules. $4 billion.
Yes, exactly.
Hi, Eli Bixby. I'm from Cradle. I really like your comments around we need something like PDB for protein-protein interaction. I think given that it's a combinatorial space, it seems entirely possible that that's just, we can't get to the necessary data density. But if we can get there, we certainly need a good library-on-library method, which seems to be evasive, to say the least. Is there anything that any of you are using that seems effective in this space?
That's a tough question, right? So
That's a tough-
library on library to
I
generate.
mean,
sort
I'm
of
not using
large-
it, but probably we all heard of A-alpha bio, and maybe that's not an exact solution, but that's an approach that is definitely worth paying attention to. I don't know they're here in the meeting. I know you interview them, right? Yeah.
All right, so right up against the hour, Paul, we'd like you to give the last question here.
Thanks very much. Again, congratulate the panel for really terrific discussion. One problem that was alluded to earlier was around immunogenicity prediction. I know that's an incredibly complex topic, and I'm wondering if anyone on the panel would care to comment further on that.
Pamela, how are we going to solve that problem, or where should the field focus their future effort in the area of immunogenicity?
Well, I think, Paul, you should probably help us answer that question.
That's right.
Thank you for a wonderful review on the matter, by the way. I think there are only so many preclinical experiments you can do to be able to de-risk immunogenicity. I had hoped, and I continue to hope, that we would be able to collect data across the industry for immunogenicity and try to solve this problem. I think this is a significant problem in the biologic space, and I think it'll get exacerbated by making molecules using machine learning and AI. I've always had this dream that perhaps we could have a consortium that allows companies to be able to share their data openly. You could blind the name of the molecule, but to share the data in a manner that allows people to learn from actual clinical findings of ADA. And I think there are some efforts in that direction, but we need to push this a little harder. But the typical approaches of mapping the particular epitopes using MAPS assays or using in silico predictors and then testing them in T-cell antigenicity assays and DC uptake assays, and those are pretty well established and people are using those. It would be nice to see if people could do that at a scale that could then be able to train predictors for these individual attributes and maybe use those and correlate those to clinical outcomes. Again, this is going to be a huge lift, but I think this needs to be done to be able to even begin to try to understand immunogenicity.
All right, well, with that, let's thank the panel one more time.